Thank you for coming today. So the work I'm going to present today is some work that I did a few years ago that was funded by the National Institute of Aging to help us answer the question of the role of microvascular blood flow in healthy aging. And that really takes us to the question of, we all know that exercise enhances microvascular blood flow. And we know now that mixed meals enhance microvascular blood flow. And I had taken it back one step before that, which was my interest in insulin-mediated microvascular blood flow. So I'm going to talk to you a little bit about that, the progression of that work, and hope that at least as we all grow older that we look more like this individual than this individual, and how can we do that. So let's take a first look at a window into healthy aging. So what I was really interested in at this time, and still am, is how aging influences skeletal muscle's hemodynamic response to anabolic stimuli. And that in particular is exercise, a mixed meal, insulin, and nitric oxide. Those were the primary areas that my lab was studying at this time. And whether skeletal muscle's hemodynamic response to these anabolic stimuli is important for healthy skeletal muscle as we age, to maintain a healthy skeletal muscle. So there was some work done back in about 2000. This particular paper was 2000, but a few years before that and a few years after that, there was this concept of anabolic resistance as it relates to aging. In the sense that skeletal muscle of older individuals, if you challenge it with a protein-based meal, mixed meal, or even just a solid protein supplement, you can see that in a fasted individual, between young and old, there's no difference in muscle protein synthesis. This is a stable isotopic measurement in percent per hour. Where we infuse a stable isotope. We take some blood and skeletal muscle biopsies, put that through a mass spec, and we're able to model it and get an actual rate of skeletal muscle growth, if you will, or protein synthesis. If you challenge with a meal that contains protein, what you see is that young have this great ability to stimulate skeletal muscle in response to a protein-based meal. However, older individuals that are also healthy, similarly healthy, are not as capable of doing that. And we didn't know why. We initially thought that maybe it was a blood flow related aspect, but we, like most scientists, started at the top and said, let's look at whole leg blood flow or total blood flow in response to an exercise bout and other interventions, and let's see if whole leg blood flow is the answer. And so what we did was a study where we took individuals 65 and older, put them on a treadmill, inserted femoral lines, a femoral arterial and a femoral venous line, so we had the leg as a controlled environment. We were able to infuse amino acids into the leg, and of course what goes in comes out, and we're able to measure what goes across. So how much of that goes into the femoral artery and comes back out the femoral vein versus what's deposited into the skeletal muscle. Sort of a crude examination or a crude explanation of a very precise examination, I should say. And what you see here is at rest, there's really no difference in total leg blood flow between old and young, nor at 60 or 180 minutes of post-exercise. This is exercise blood flow measured with indocyanine green dye dilution, which is a technique that we can do if we have femoral lines placed in those particular patients. So if it wasn't total leg blood flow and exercise in and of itself between young and old, total leg blood flow was the same, then what was it? What is it that's causing this apparent discrepancy between the anabolic potential of older and younger skeletal muscle? Several of my colleagues that were in Australia were looking at this particular question and had moved to University of Virginia, and my group went to the University of Virginia and learned this this particular technique so that we could start measuring microvascular perfusion and in that case, nutrient perfusion. So we're really interested in what's happening to those nutrients at the level, say, of the capillary, whether it's being exchanged, whether they're being picked up and used for an anabolic process. So today we're going to talk about, is peripheral vasodilation altered with aging? What stimulates vasodilation in the periphery, and is it important for nutrient stimulation of muscle anabolism? Does enhancement of this peripheral vasodilation and muscle microvascular blood flow actually restore the sensitivity of skeletal muscle with aging, or does it just stay roughly the same? And what role, if we have time, what role does the muscle interstitium play in this process? So just as a brief reminder of the vascular tree, so we initially started by looking at the resistance arteries, looking at the influence of total blood flow, and then we really wanted to work our way down from the terminal arterioles through the capillaries. We were more interested in controlling nutrient exchange. If we provide the nutrients such as amino acids to the skeletal muscle and we enhance perfusion to presumably get more of those amino acids that are anabolic to skeletal muscle, to maybe closed capillary beds or something like that in response to exercise, then we were more interested in what's that response going to be. This is a technique where we're able to essentially measure microvascular blood flow in sort of a pre-post comparison state. So you have to do it at baseline and then in response to something, okay? Something that you know is going to stimulate it. It's called contrast-enhanced ultrasound. This is an example of some work by Jonathan Lindner, who is at the University of Virginia at this time. He's a cardiovascular surgeon. He has taken this to a whole new height and is using these microvesicles to deliver drugs and vectors and all sorts of things in a targeted way. In this case, all we're doing is infusing these microspheres, which remain intravascular. They're echogenic when they're exposed to ultrasound, meaning that when you put ultrasound to them, they pop on the screen, just like if you wanted to see what your baby looks like. And then they have similar flow properties to that of red blood cells. And that's very important because the nutrients, as they traverse through the blood cells and the capillaries, we wanted it to have a similar concept to that of the flow properties of erythrocytes. Use a particular ultrasound machine, and then we destroy those microvesicles, and when we destroy them, we watch them from the point of destruction to refilling of the capillary beds. And again, as I said before, this is recommended really more for a pre-post comparison of some sort of intervention. So here's how this works. You infuse a constant infusion of these DEFINITY microbubbles. It's more of a pulse bolus type scenario. You burst them at time 0, and you're with the thought that you're gonna establish a curve. So you pulse at time 0, 0.1 seconds, 1 second, 5 seconds. Each time you see we're getting a little deeper fill into from the large vessels all the way down into the microcapillary vessels. 10 seconds, 20 seconds. Now we're establishing our curve from 0 across up to 20. And you can even go even longer depending on what it is that you are anticipating your curve is going to look like based upon your intervention. So if it's a very quick change, say with nitric oxide versus exercise, you can adjust your curve accordingly. And what we get from this is a curve fit of video intensity to this pulsing interval. We get an A and a beta, and from that we can assess microvascular blood flow. So this technique, again, is a pre-post assessment. So what we would do then is we designed a study where we had older individuals, again, doing endurance exercise for 45 minutes at 40% of their VO2 max. All we wanted these older folks to do was walk on the treadmill at a good clip with a slight grade. And we did all of these measurements again, indocyanine and green for whole leg blood flow, CEU for the microvascular blood flow. We did some microdialysis sampling. We put some stable isotopes into these individuals and did muscle biopsies across time, and of course took blood samples. And during this, we had a resting period, this short 45-minute exercise period, and we, in the post period, we gave them some amino acids to see if we could stimulate anabolism, and of course measured them during this post-exercise period. This nitric oxygen donor scenario up here is for the second study, because we did a second study that was nearly identical to this with a donor instead of exercise. So what did we find? We showed that microvascular blood flow is blunted at rest and after endurance exercise in older individuals. Now keep in mind, we had shown that whole leg blood flow was the same between young and old, but if you look at the resting period between young and old, that there's a significant difference in microvascular blood flow in the young and old at both rest and 60 minutes post-exercise. Not good news if you're getting older, because what that means is you don't have the same anabolic potential as an older person that you do as a younger person. So it kind of explains why grandma starts walking like this as opposed to more upright and having a good muscle mass. So when we looked at this then using our stable isotope analysis, and we modeled the different efficiencies and transports of these amino acids going into the leg, out of the leg, into the muscle, and so forth, what we saw was that if the young are the lower group here, this is outward transport. So in other words, less of what was given in terms of the amino acid was coming out in the young, which meant more was staying in the muscle. Versus the old, we had a much greater outward transport efficiency. So it's a backwards thinking, in other words. You don't want to have more amino acids escaping back out. You want to keep them into the muscle so that you can have normal protein turnover. We also showed that and this is the reverse side of it, that the young then had a significantly greater synthetic efficiency capability. So what you provided them, they were much more efficient at turning into an anabolic stimulus, if you will. So if we then take that exact same study and we recapitulate it with a nitric oxygen donor like a sodium nitroprusside, we infuse that into the femoral artery, and we essentially take everything from the femoral artery down to the toes, and we open up all the capillary beds with nitric oxide. We can effectively normalize the scenario. So this is the young photo of the ultrasound when you've given a nitric oxide. So this is just a qualitative examination. And you can see down here in the old, we have a similar response. And that's what we showed here, in that overall microvascular blood volume is very similar between the young and old when you give a nitric oxide donor. So being able to give something like Viagra, for example, which we know stimulates perfusion, might actually have some muscle growth potential. So more to come later, just to stimulate some thought. So does enhanced skeletal muscle vasodilation translate into greater muscle anabolism? I've kind of already given away the answer to that, but we found that the nitric oxide donor, sodium nitroprusside, restores muscle protein synthesis in healthy older men and women. This is again a stable isotope of phenylalanine that we infused into the femoral artery. We measured blood and skeletal muscle. We then got a muscle protein synthesis or fractional synthetic rate, which is in percent per hour, between basal and 180 minutes post-exercise. This is the nitric oxide donor with an amino acid. So the whole point is to give that nutrient stimulation along with a donor so that you're opening up the capillaries and you're providing the nutrients both, okay? And what we found, and we published this in AJP in 2011, is that we're able to pretty much normalize with the nitric oxide donor the anabolic potential of an older individual, similar to that of the young. The next step then we took was with Viagra. So Viagra is a drug that Pfizer has used wonderfully to make lots of money in men who have erectile dysfunction. And the process of that for the penis is the same as it is in the skeletal muscle, in the sense that it's a muscle perfusion enhancing process. And in skeletal muscle, we showed that sildenafil strongly enhances muscle protein synthesis. This was an unintended finding. We were actually conducting this study to look more at a proteomic profile in these patients for a fatigue component, because we were interested in the role of sildenafil or Viagra in reducing skeletal muscle fatigue with aging. And that was the study. We took skeletal muscle for a proteomic analysis, and at the same time while we were there, we went ahead and ran our muscle protein synthesis with stable isotopes, and we found this huge difference. This is along the lines or the magnitude of what we would see if we gave a testosterone into the same individual. And so we were astounded. We then followed up, took a look at our proteomic results, which we then published both side by side, confirming that myosin heavy chains, the actins, and many of the things that are responsible for showing an enhanced anabolism were upregulated, or the expression of those were enhanced in response to sildenafil. So you can imagine Pfizer was very interested in these results. So what stimulates vasodilation in the periphery and is important for nutrient stimulation of muscle anabolism? This work was originally started, as I said, from this group at the University of Virginia. Michelle Keski, who is now back in Tasmania, taught me to do some of this contrast-enhanced ultrasound work. She, on the other hand, was more interested in its role in diabetes, with the fact that if you could enhance perfusion, perhaps you could alter how insulin mediates glucose uptake. And so that was more of her interest. And so she did a study where she looked at what happened to your microvascular perfusion in response to a mixed meal. So there was both carbohydrate and protein as well as fat. And she showed that a mixed meal increases not only a large vessel flow or brachial artery flow, but also MBV, which is the capillary blood volume, microvascular blood volume. But you can see that there was a difference in how the lean responded at the 120-minute time frame to the obese. So the obese did not have as nice of a response. It was much more blunted. So that has repercussions then for obese individuals who are also diabetic, who you want to try to help manage glucose metabolism. She then looked at insulin and how it stimulates skeletal muscle vasodilation and femoral artery flow. And again, she showed that insulin, which we all kind of thought at this point, insulin is a very good mediator of capillary blood flow. The comparison here is saline versus insulin. You can see at 30, 90, and 120. She has a nice microvascular response, as well as over here, though, in the femoral artery flow, you only see it with the insulin as you get into the longer time points. So you don't have that ability looking at larger flow, vessel flow, to be able to pinpoint what's happening. So a group of my colleagues at UTMB at the medical school, we published a paper in diabetes where we were looking at a similar study that we had done with the nitric oxide donor, sodium nitroprusside, except that they were adding insulin into the mix just to see what would happen. And you can see that there's no difference in this microvascular blood flow at the fasting state in either one of these, whether it's perfusion or synthesis. But when you give insulin in this, you see this beautiful response in muscle perfusion. So not a big surprise. Thankfully, insulin's there to rescue the day so that if we need to have an insulin-mediated control of our blood sugars and other things, then it's there to help. So, okay, if we can do it with a drug, can we do it with exercise? So this group, my colleagues looked again at aerobic exercise in controls and exercise patients. Same result. Essentially, exercise had a similar response. These studies, I believe, were in young. No, these were in elders. So they saw with a harder exercise this much enhanced increase in muscle perfusion as compared to what I had shown at the 40% VO2 max. So we get a good response with muscle perfusion and muscle protein synthesis in elders in response to aerobic exercise. So there's some more hope. It's not just going to have to be drugs. So we know from this data then, if we look at qualitatively young versus old, in the basal state, the young don't have quite the perfusion that the old do not have quite the perfusion that the young do. If we exercise them, give them insulin or mixed meals, young still have a higher response, but we know we're capable of stimulating that with exercise and mixed meals in the right circumstances. And we know from these data that nitric oxide donors are the best. So Viagra, sodium nitroprusside, those types of drugs that we can use to open up the vessels. Here are the references if you need them for some of these studies, the exercise, insulin, mixed meal, and the nitric oxide donor studies. For your reference if anybody wanted to take a photo of those. Okay, so we've looked at whole leg blood flow. We've looked at microvascular perfusion of the capillaries in skeletal muscle. But what about that in between? So when we eat a meal or we give amino acids, goes into the blood, crosses across from the vessels across into the muscle. Well, there's a period in there in the interstitium. We don't know what happens. What role does that interstitium, the skeletal muscle interstitium, play in this process? It's begun to be further elucidated in the glucose world, glucose insulin world, in terms of the myocytes. Lots is known, but less is known what happens with aging in the interstitium with amino acids and some type of enhancer. So the skeletal muscle interstitium we know can help regulate blood flow. We know that it is involved in the transport and passage of substances from the capillaries to the myocytes. There's a role of regeneration, and there's also more evidence now that there is an intercellular signaling that is very important in skeletal muscle. So my lab worked with Bob Hickner from, blanking where Bob Hickner's from right now. Anyways, he came to my lab, we collaborated, he taught us how to utilize the microdialysis ethanol technique. My lab was already using microdialysis probes, but this ethanol technique allows us to actually measure the blood flow of the interstitium. So we know what the whole vessel flow is. Now we can measure the interstitial flow, and we know what the capillary flow is. So you get a local nutritive exchange measurement. You infuse, just like if you think about the concept of any of your family members that have to go in to have kidney dialysis, it's exactly the same concept. You're putting in a perfusate and you're collecting a dialysate. You analyze the dialysate for ethanol using a fluorometric method. And this is the only thing I was mad at Bob for. When he published the first paper, he put it as an outflow to inflow ratio. And he should have just flipped it, because by leaving it as an inverse, when you look at the figures, everything inverse, so the greater the inverse, the greater the flow, as opposed to if he had flipped the ratio, it would have helped those of us that were trying to present the data. But the bottom line is we now had a wonderful way using these probes to measure, to measure flow of the interstitial substrates. And while you're measuring flow, you can also collect whatever is in that interstitium. So whether it's amino acids, any concentrations of insulin or cytokines, anything that might be positively or negatively influencing skeletal muscle. How are we doing on time? Okay. So we looked at interstitial flow at rest and after endurance exercise, and we found it to be lower in the older men. So if you, again, this is, you're seeing it higher here, but it's inverse. So the older men at all time points rest post-exercise at 60, 120, and 180 was lower in the older men. So we now have, we saw no difference in total flow between old and young. We're seeing a lower flow in the interstitium between old and young, and a lower perfusion between old and young. So we're digging down a little bit deeper and can see that there might be some reasons for this blunted sensitivity of muscle protein synthesis to amino acids after exercise or in response to other potentially anabolic agents. So not to get too deep into the weeds, but again, we always, it's very easy to infuse stable isotopes into people. A little bit more difficult to get biopsies, but if you get biopsies, you can get blood, and if you get blood, you might as well put femoral lines in. You get femoral lines, then, you know, put them in bed and pay them a lot of money and tell them thank you for your time, and we get lots of people coming back for this because they love the exercise components. They love to know exactly what's going on at all levels of their muscles, so they really get into this process. And what we showed modeling our stable isotopes is that the synthetic clearance of the old is, in this case, it's not inverse. The synthetic clearance is lower in the old than it is in the young, not good. We don't see any anabolic clearance issues that were significant, but you could begin to see some differences here at the 60-minute post-exercise. And then we see overall synthetic efficiency between young and old is slightly different in the post-exercise period in the old, excuse me. What does this really mean? Well, if we separate out arterial and interstitial concentrations and we measure what's going on between the young and the old as it enters into the large vessels and then traverses across the interstitium before it gets to the muscle, and we look at those particular amino acid concentrations, and we look at the amino acids that we know are highly anabolic to skeletal muscle, and that would be things like leucine or whey protein, which has lots of leucine in it. We can see that in the young, we see what? Lower levels of leucine. What's happening here? So this was a big shock to us. In the artery, we saw that the old actually had some higher levels of arterial leucine than the young. Okay, well that's strange. In the interstitium, we saw the same thing. If you look at the phenylalanine, it's the same story. Okay, so we were a little bit perplexed. So why are the old, why do they have more of these anabolic amino acids in their blood or in their interstitium than the young? Because for many, many years, even my doctoral advisor, the concept was that it was an amino acid availability issue. Okay? The reason that we became frail when we got old was that we didn't have enough amino acids with age in order to sustain sort of this anabolic potential. But in fact, what we're showing here is that there's plenty. We're just not doing something with them. Okay? There is truly a resistance at the level of the muscle. There are plenty of amino acids, and in fact, there's more in the old than there is in the young. But we don't know, we at this point didn't know what that's really meant. So if we look at the overall compilation of arterial essential amino acids, so all the key ones, leucine, isoleucine, valine, all the ones we know that we like to have for stimulation of muscle protein synthesis and nutrient exchange at the level of skeletal muscle for anabolism. We look at those, again, same story. The old have plenty of amino acids, and in fact, statistically more than the young. Same thing here, if we look at the compilation of all of them. So it wasn't just those few major amino acids like leucine that we know to be highly anabolic. So then if we look at our story of, well, what's happening to the rate of appearance or the intracellular rate of appearance of these particular amino acids, we can see again, there is the old maintain this intracellular rate of appearance of amino acids, which means that the amino acids are there. It's not a rate limiting step in this anabolic process. Yay for old people. How does this correlate though? So if we look at the correlations between, so we have protein breakdown is in the open circles, protein synthesis solid, and net balance is the half. We see that there is a good correlation in the essential amino acid group with net balance. So what is that telling me? Net balance means protein turnover. What you want to have healthy muscle is protein turnover. You want breakdown to occur, you want synthesis to occur normally, right? And we see this normal correlation is occurring with all of the essential amino acids, both with the plasma and the interstitium. So this sort of confirmed that we still have the machinery in the skeletal muscle that's capable of doing this, but we need to help it along a little bit. So what we found then was arterial concentrations of several amino acids were higher in the old than in the young. This was not due to an elevated overall endogenous plus exogenous rate of appearance as the RA in the old, sorry to use these old terms that, these kinetic terms that don't make any sense to other people, but essentially the rate of appearance of these amino acids in the old, in the whole body was similar between young and old. So that wasn't our problem. And the interstitial concentrations were similar, and they were highly correlated. So with that of the plasma. So it's not that. For 20 years, I was taught in my postdoc, that's what it was. We don't have enough amino acids, it's rate limiting. Well, now the story is there's some blunted anabolic sensitivity. Basal muscle protein metabolism we found overall was similar in the young and old. Mixed muscle protein fractional synthetic rate, which is just muscle protein synthesis, a fancy way of saying it, was significantly higher post-exercise than during rest without age-related differences. So both are able to exercise and increase their muscle protein synthesis. That's good news. Net balance of muscle protein improved in the post-exercise period. That's good news. So the young and the old are capable of getting this anabolic machinery working, but we know that there is still something going on in the old, okay? And we found this moderate to strong correlation between net balance, which is that muscle protein turnover, that healthy factor that we want to have happen in everybody in skeletal muscle so that we can maintain a healthy skeletal muscle. So we then looked a little bit deeper and we started looking at markers of metabolic and oxidative stress and whether or not they were higher in the muscle of the old versus the young. And we found a few. So we found a few things that might lead us down the road to say what's different. If it's not amino acids, then what is it? So there's this, for many years, this concept that the old have this sort of subclinical inflammatory process going on, and we can't help it. I mean, you just get, the older you get, things start to creak, things happen, you get this subclinical inflammatory process going on. Maybe you start having some mild arthritis, maybe some of these other inflammatory diseases that start to crop up that are age-related diseases, and these are subclinical inflammation. You're not going to the doctor because you're sick, but you start noticing these things in your joints and other things, right? Well, what happens is that it is truly a clinical scenario. You get things, cytokines and other things, ammonias and cysteines and 3-methylhistidines and all sorts of other markers in the blood and in the interstitium that tell us that we are inflamed. But wouldn't, if you went to your physician, he wouldn't say, you know, you're inflamed, you need to do something about it. Of course you're inflamed, you're older, that happens. But what do we do about it? So we found ammonia to be a factor. We found that there were increased glutaminase activity going on. We found higher levels of 3-methylhistidine which is a marker of myofibrillar protein breakdown. So maybe there's some higher muscle protein breakdown going on in the older compared to the young. And then we found this cysteine, which is the oxidized form of cysteine. And we know this, that within this interstitium and within the blood that we see these things, and it tells us that we're old, makes us feel old. So what's the moral of the story here? At rest, we know that older men exhibited significantly reduced skeletal muscle, microvascular, nutritive flow compared to younger men. After exercise, we're able to somewhat enhance that, but not fully. It doesn't return to this absolutely normal response. It's still mildly blunted. And if we add amino acids, we get an even better response than if we just do the exercise alone. So that substrate is important. It's not the 20 years of giving amino acids, or the 20 years of where we thought that amino acid availability was a limiting factor with age was not the issue. Yes, if you give amino acids to young and old, it grows muscle. But it's not something else, the stomach or something else is picking up those amino acids. There's still plenty there at the level of the muscle to do what it needs to do. But something else, perhaps maybe an inflammatory scenario, oxidative stress, something like that. We do know that if we pharmacologically vasodilate with a nitric oxide donor, we can provide a similar microvascular flow response. And that then can help normalize this muscle anabolic between young and old, which is important because maybe someday if you're in the hospital, that's what we're going to be doing as you're in a bed rest scenario where you're having higher levels of muscle protein breakdown. So aerobic exercise, insulin, mixed meals. So whatever you had for lunch today, your perfusion went up right after you ate lunch today. Good for everybody in this room. Tell your grandparents, tell your parents, keep eating 3 squares, and it will help even if they're not exercising. And of course, nitric oxide donors are capable of increasing this vasodilation and overall muscle microvascular flow. We believe that if you can enhance microvascular blood flow, whether it's through exercise or some other means, you can facilitate substrate utilization. Whether it's you're trying to enhance that in an athlete to help them grow skeletal muscle to enhance their sport performance, or whether you're trying to keep your grandmother out of a nursing home. We know that protein turnover, normal protein turnover in skeletal muscle is a good thing. So anything you can do to enhance muscle protein turnover, to not necessarily enhance breakdown, but breakdown is important because from breakdown we liberate amino acids, right? When you break down skeletal muscle, it liberates amino acids, and if we can reutilize those amino acids back into the skeletal muscle, then that's a positive outcome. So protein turnover that occurs in response to exercise is a good thing. The breakdown component that occurs in response to exercise is a good thing. You just have to make sure that you provide the substrate in order to continue to grow it. So these data underscore the importance of muscle's microvascular hemodynamic response and the interstitial milieu in healthy aging. So I'm happy to, not a big Monty Python fan, my husband is, but I'm not a big Monty Python fan, but it just seemed appropriate since everyone seems to be looking for that holy grail to be a better athlete, not get too frail when they get old, and so forth. So I'm happy to take any questions that you might have.
I know you weren't necessarily looking at like older individuals necessarily, but do you think there would be a different framework if the individual had been like regularly resistance training all throughout his life? Like I'm assuming, obviously would have better benefits, but nevertheless, the people who don't have that kind of training history, would you then recommend that they have like forms of Viagra, I guess, to help better facilitate?
Well, so something something I should have said is that these are all healthy older men.
Right.
These are men that are 65 on up, that were still out mowing their lawns every week. They're walking, they're running, they are exercisers. They may not be bodybuilders per se, but these are all very functionally healthy older men, because we wanted to make sure that we had a functionally similar group to the young. Now the young, in fact, were more couch potato-like in the sense that we didn't take athletes that were exercising every day, or we took people, I think, that were exercising no more than 3 times per week at a moderate level, because we were trying to match the groups better. So do I think maybe that there's still some anabolic resistance in someone who has trained their entire life? Yes, I do. Because I think, unfortunately, oxidative stress and lots of other things, our capillaries become more leaky, all sorts of things happen with age that we can't control even with exercise. We can only try to tamp them down a little bit. So I do think it's still happening. I think that it's the good news from this is that even there is a way to correct it, right? And the story would be that if you can do anything that enhances your blood flow post-exercise, blood flow response, that's a good thing. And if you can toss in a substrate in the post-exercise period, which all these athletes know, that if you toss in some protein at the right at the tail end of the workout, guess what? You grow muscle a little bit better and get a little bit bigger. So, yeah.
I'm kind of going to go the other direction, away from resistance training. Did you do any baseline VO2s maxes for any of the individual?
We did some baseline VO2s to establish their 40%, but we did not do a post VO2 because we really just needed it to establish the 40%. Are you asking, do I think whether VO2 max
I
would
would think that, especially in the older individual, that the fitter you are, the more protected your endothelial cells would be, which would produce more nitric oxide, which would help with perfusion.
Right.
So, have you seen any of that?
So you deal with that with the reviewers, right, when you send the paper in. Because either way you look at it, you can argue, well, you should have gotten really unhealthy old and some healthy young. Okay, well, that's one question, right? Could have gotten, we tried to basically match the young and old with every aspect other than maybe, with their exercise aspects other than maybe their age. We didn't allow anyone to come in that had any recent illnesses, no cancers, no nothing. These were as healthy as you can get for a 65 plus person. We also limited the upper age range for that very reason, because there were opposite arguments that could have been made in either direction. And so we kind of fought those in the process of the paper, even with how we selected to ask that question, which was we really wanted the young and old to be similarly healthy, and really age just to be the major factor. So that was why we selected it as we did. I see where you're going, though, and I think that's just a slightly different question from what we were wanting to ask.
Future studies to
Right.
investigate that.
Now we
answer every question in one study.
Now the interesting thing with the resistance exercise component is as you know, with this whole pumping action of skeletal muscle, when you're doing aerobic exercise, you get less restriction of the skeletal muscle with an aerobic event, right? Versus a resistance exercise, which is more of a restrictive event to the skeletal muscle. And so the evidence suggests from our lab that there are some really neat things, and there's some blood flow restriction studies that have really taken this to a whole new level, where they've seen some of the really interesting components that come out of the skeletal muscle in response to blood flow restriction. So what would be that, likening that to a resistance exercise bout. And so some of those things are very beneficial to skeletal muscle in terms of healing skeletal muscle. So there's a whole nother line of research that could be opened up along this route. Problem is these studies are really invasive. You have to do them in a hospital setting. You have to have an IRB that's willing to allow you to poke and prod old people like this. And you have to have older individuals that are willing to do it. And you know, we lived, I did the study at UTMB in Galveston, and you know, Galveston has a lot of older individuals that walk all the time, and they want to live near the beach, and so they have, you know, presumably that more of a healthy outlook and lifestyle. So, you know, we didn't have any trouble recruiting to get people to come in to do these things. Anyone else?
Some of those studies that were mixed meals, do you have information about the ratio of breakdown of the macronutrients?
So those mixed meal studies were Michelle Keske's studies, because
relative to the actual content of the mixed
yeah,
meals?
that's all in her papers. I want to say, I had that on my slide before, but I think I took it off. I'd have to go back and look. I can get you that info.
Or I can find it.
Yeah,
Give it
it's
whatever.
Keske, yeah, Michelle Keske. It's Vincent Keske. So
was there, did you take any surveys with the Sildenafil to see if any of the participants had any other comments?
yeah, so we continued to do years worth of sildenafil studies. We did some sildenafil studies with both men and women, and because once we found that it was highly anabolic, there was a great interest in it, obviously.
I didn't know if you had any
So we're,
comments that you got from
well, number one, the number one comment everybody asks me is, you know, similar to the testosterone studies that we've done, the women married to the men are always like, where's mine, right? So I give a male testosterone, and he's chasing the wife around all the time, and the wife's like, this isn't fair. I need something too. So we were kind of hoping that with sildenafil that was the case, and it had not been studied over a long period of time in women, and we gave it to older women for 3 months. And while we showed some pretty significant growth enhancements in terms of skeletal muscle, unfortunately, in some of the questions that we asked the women, we did not have any of the women say that they had noticed any enhanced sexual interest, which is actually the number one. No one cared about the muscle. Everybody wanted to know, was the wife more interested? No. The answer to that was no. We didn't get that response. So.
This is a somewhat naive question, but you referenced blood flow restriction studies. I'm assuming this is what people call katsu?
Yes.
Yeah. So when you basically you're applying a tourniquet, right? Say,
Yeah, it's like a blood pressure cuff.
so does that? So you're blocking some of the venous return, correct? Does that enhance microvascular blood flow?
Well, it has not been measured, I don't believe, that sort of post-release or the release following the constriction. It's probably would be tough to do that with the techniques that I have. Number one, we could probably do some downstream muscles, but like if you do it on the thigh, kind of limits doing any type of microvascular measurements, because you're where you need to be is where you're constricting, and that's a very quick. Once you release the cuff, it's a very quick response. And so what they've strictly done, Blake Rasmussen's group and others, Satoshi Fujita in Japan, they've looked at the synthetic response, but they haven't really looked at the blood flow response. Everybody knows that you're constricting. They know the level that they're constricting, but that sort of release enhancement, I don't think anyone's looked at the microvascular flow.
Do they find an increase in protein synthesis?
They do. Yeah, they absolutely do.
Probably have time for one more question. Liz, did you have your hand up earlier?
Yeah, no, it kind of got answered with
Okay.
one of
Yeah,
Travis's.
Yeah, no?
It was too close to Travis's question.
Okay, okay.
It was good work.
Well, I want to thank our guests for coming today. If you could thank our presenter, that'd be great.
Thanks for having me.